before we proceed to the tissue processing, you have to make sure that the tissue cassette is properly labeled as what is in the laboratory sheet, the patient lab number. So for example, for this case, we need to label our tissue cassettes, with the label written on the form as well. Then we proceed to tissue processing. Okay, after the grossing, we submit the tissue for tissue processing. This is the tissue processor. This tissue processor will run overnight to finish the entire fixation of the tissue samples. So this is how we operate it. The first station will be the formalin. Okay, you put the tissue basket here containing the tissue, okay, into the tissue basket. This is the tissue holder. Put on the tissue basket with the soft tissue inside. Okay, put it down, and then you start the processor by pressing this one, and you allow the machine to process for 16 hours. So tissues will be finished, if processed by 2 o'clock, will be finished the next day by 7 a.m. After tissue processing, this would be the end, the last station of the tissue processor. Okay? So we remove the tissue out of the paraffin wax. Okay. Fill this cassette molder with melted wax. So we go here. Fill this with wax. Bring it to heat so that the surface will not be solidified. Take your sample out of the wax, okay? Transfer it to the melted wax here. Position your sample with the cut surface facing down and cover it with the label of the sample from the patient. So hit this tissue cassette warm, make it warm, and place it on top of your sample. Wait for your sample to cool down at room temperature for at least 30 minutes before placing your tissue block inside the freezer for at least 20 minutes. After allowing your sample to cool down at room temperature, this will be brought inside the freezer for at least 20 minutes, and then you will be able to remove your tissue block from the tissue molder when it's very, very cold. Now it's time for sectioning. Now, the first procedure that we are about to do with our tissue sample is to trim off the excess wax on the tissue to expose the entire tissue sample. Okay? We usually section our soft tissues at 5 microns. This is the ideal thickness of soft tissues for H&E stain slides. After you have removed the excess wax from the soft tissue, we will begin to collect tissue ribbons and put them on the slides. Okay. As you can see, tissue ribbons are coming out. So what we should do is to collect each tissue ribbon Be very gentle when collecting the tissue ribbon to preserve the soft tissue inside it. So we put this in a cold water just to allow for the tissue ribbon to be straightened. Then we fish it out using the glass slide. Transfer it to the water bath set at 37 degrees. You will see that the tissue ribbon will be entirely smooth before you put it on the glass slide. So allow at least 10 seconds to straighten the entire tissue ribbon and the tissue itself before we collect it and put it on the glass slide. Make sure to label your glass slide using a pencil, because pencil will never be erased when this glass slide will be placed in the tissue processor later on. After the tissue processor had finished the program, remove the entire rack of the stained slide and proceed to the last procedure, which is called the mounting and labeling of slides. For the mounting and labeling of slides, what we do is we remove the slide from the last station, clean the surrounding of the slides together with the tissue, apply the mounting medium, this is called DPX mounting medium, onto the tissue, and then cover it with cover glass on top. This is an example of a glass slide covered with the cover glass with the H&E stained sample on it. Thank you.